Hello, good afternoon. I am Badaró Mendonça, PhD student at FIOCRUZ, supervised by Tatiana Chile and Mariana Vagabi. The study presented today is entitled Identification and Validation of Genes Candidates as Targets for Treatment and Diagnosis of Breast Cancer. Breast cancer is the most frequent cancer among women, affecting 2 million women each year. In even more alarming numbers, 685,000 women die from breast cancer only in 2020. There are 5 standard breast cancer treatment options, but despite the clinical advances, the treatment has some limitations, such as systemic toxicity, resistance to drugs, and debilitating side effects for patients. So far, I have talked about numbers and point out some problems, but this image shows the truly reality of the patients. They suffer in the short, medium, long term due to the current therapeutic approach with a lack of specificity. So the big question is, how can we improve quality of life and minimize the side effects? So membrane proteins appears as an interesting strategy for target cancer therapy. So our main objective is to identify targets that are overexpressed in tumor cells and that have low expression in normal cells, bringing more specificity. So to attain our objective, samples of transcriptome of tumoral and non-tumoral breast cancer tissue were downloaded, and we also downloaded other non-tumor samples. Then the identification of overexpressed transcripts was made, and this stage 4 proteins were identified, which peptide is pending, and finally protein expression was validated by immunofluorescence. We analyzed the expression profile of the 4 identified transcripts. We can observe that the breast tumor samples in blue present an overexpression in all graphics compared to the breast control in the black one, and also in comparison with the control samples from other tissue. Next, we explore either there was a difference between the gene expression profile of the target genes in relation to the molecular subtypes, and we observe that the gene D has significant expression in group luminal compared to the control, whereas the gene C has show high expression in the group luminal and R2 compared to the control, while the gene A and B are expressed in high levels in all groups of the 4 molecular subtypes compared to the control, including the triple negative. That is an interesting result because the triple negative is the most aggressive with the worst prognosis. So these two targets can be great to diagnose, such as target therapy for this kind of cancer. We also characterize the targets as a biomarker for breast cancer, analyzing the sensibility and specificity through the ROC curve. In the chart A and C, we have sensitivity and specificity greater than 80%, and the B and D, we have the greater than 70%. In our chart, we have the area under the curve above 0.7. So this data indicating optimal levels for a tumor biomarker. Last not least, we validate the expression profile of protein by immunofluorescence assay. And the first column is the non-tumor cell line. Then we have 3 tumor cell lines that contemplate the different molecular subtypes. As we can see, the image shows an increased staining of the 4 proteins in the tumor breast cell lines compared to the weak or absence staining in the non-tumor cell line. So this data corroborates the result obtained with the RNA expression. As a brief conclusion, these four membrane proteins could be considered for clinical relevance as a tumor biomarker, definition of tumor-free, target delivery, and for monoclonal antibodies and or epithemers. So I would like to thank the organized opportunity to present my work. I would like to thank my work team as well, and thank you very much for your attention.